Hello, everyone, and welcome to today's webinar titled Solutions for Accelerating the Maturation of Cancer Vaccines. This webinar is a part of the third annual event in the Immuno-Oncology virtual event series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Melody Chang, Segment Marketing Manager at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Melody, you may now begin your presentation.
Hello, everyone. Today, my topic is solutions for accelerating the maturation of cancer vaccine. So there are three parts. First one, I'm going to introduce you guys some the typical vaccine types and mechanism. And the second part, we will discuss linear mRNA vaccine and circular RNA and saRNA as alternative approaches. And the last part, we will see the importance of neoantigen peptide for personalized cancer vaccines. So first part is typical vaccine types and mechanism. So by format, the vaccine types can be classified as three generations. The first generation is virus-based. The second generation is protein-based. And the third generation is nucleic acid vaccine and viral vector vaccine. So for the first generation, the virus-based vaccine includes inactive vaccine and live attenuated vaccine. So basically, they just use the virus itself, either inactivate it or make it live attenuated. So it's weakened. And the second generation is protein-based. We just use the subunit part of the virus. So it's extracted from pathogen or constructed by gene engineering. Usually it includes peptide vaccine and recombinant protein vaccine and virus-like particles. And for the third generation vaccine, so for the nucleic acid vaccine, usually we use the nucleic acid, so includes DNA and mRNA, so just the genetic material. Or we can use a virus as a vector to take the nucleic acid from the pathogen into the cell. So that's the three generations. And let's look at the mechanism. So generally speaking, there are three steps. First, we introduce a kind of vaccine into the cell, and after intracellular processing, what we need is the peptide. The peptide is gonna be used to stimulate the immune cells So there's gonna be immune response, including cellular immunity, which means some cytokines will be released, and humoral immunity. So antibody is gonna be produced. After this immune response, the third step is the infected cell specific lysis. So that's the way to check whether the vaccine is effective or not. So that's the whole mechanism and steps. And we can see here it's more clear, like different types of vaccine, including viral vector vaccine, DNA vaccine, mRNA vaccine, and recombinant protein vaccine here. And also we have peptide protein or peptide vaccines. Okay. And the next part, we are going to look at the pro and cons of different vaccine types. So for the virus-based vaccines, generally speaking, the advantage is it's going to have the stronger immune response. But the disadvantages is like, for example, the live attenuated virus vaccine, it still has the risk of residual virulence. So it's kind of dangerous. And for viral vector vaccine, it's safe and effective. But the effectiveness might be compromised by pre-existing immunity to viral vector itself. And for subunit vaccine, it's highly safe, but it needs the adjuvants to increase the immunogenicity. And for DNA vaccine, it's safe and well tolerated and stable under room temperature and highly adaptable to new pathogen, but it has lower immunogenicity and a difficult administration route. And still, most importantly, it has the risk of genomic integration. And the last one is mRNA vaccine. It's simple and fast to design in response to virus outbreaks. We have experienced it. It has successful case. And the mRNA vaccine has launched onto the market. It can induce both humoral and cell-mediated immunity, and highly adaptable to new pathogen because it's a platform-based vaccine. But the disadvantage is some vaccines require cold chain storage and supply, and the long-term effects need to be evaluated because it's the third generation. We still need to see the long-term effects. Okay, we have understood the vaccine types and the mechanisms and pro and cons. Next, let's just spend some time on mRNA vaccines because it's most popular recently. And we're going to compare linear mRNA with the advanced one, circular RNA, and saRNA. So see these two alternative approaches, and we're going to compare them together. Okay, so this one shows a comparison between these three kinds of RNA. We compared the structure, ribosome recruiting, ORF, UTR, poly A tail, protein expression, half-life, immunogenicity, dosage requirement, and novelty. Okay, so let's see the structure part. So basically, we can see that this one is like a circle one. It's a closed loop. No, so it's eliminating the mRNA exonuclease degradation. But these two, it still has this mRNA exonuclease degradation, and it's both of these two are linear. And also they have this cap, but this one doesn't need a cap, but it needs the internal ribosome entry site for translation. And for the ORF part, this insertion is below 20 kb, and this one is below 6, and this one below 4. And UTR part is also different. And this one, circular one, doesn't need poly A tail, but these two need it to for stabilization. And let's see the protein expression. So, self-amplified RNA or saRNA has the strongest protein expression, and the circular RNA is stronger. And this linear one is just strong. We give it strong. And for the expression half-life, saRNA has the longest half-life, and this circular one is the second, and the linear is the shortest one. And for immunogenicity, number 1 is saRNA, number 2 is circular RNA. And immunogenicity for linear mRNA is the lowest one. And dosage requirement, of course, self-amplified needs the lowest, and the second one is circular RNA. And the novelty, so linear mRNA is the traditional one, and these two are the new ones. Okay, from this picture, we can see that mRNA needs the less amount, but the expression is the highest. Next one is circular mRNA. The last one is linear mRNA. Okay, so let's first check more about the mRNA vaccine because it's the traditional one, relatively speaking. And this is the 5 parts of mRNA, 5 elements that we can adjust to have a better performance for mRNA. So for this cap part, it's affects the capping efficiency could affect the immunogenicity. And if we adjust the UTR part, it also affects the protein expression and the stability. And for the ORF part, if we modify the nucleotide, so it can reduce the recognition by PR. And if we optimize the codon, it can help improve the protein expression. And the length of poly A tail will affect the stability of mRNA. So all these are the elements that we can adjust, and these are also the key considerations when we try to find outsourcing. And the GenScript have the corresponding solution to these considerations. So for the template of the mRNA, which is the plasmid DNA. We have plasmid DNA integrity guarantee. And also for gene synthesis, we have the codon optimization process. And for poly-A tail quality, our way is to build poly-A into plasmid. And capping, mRNA capping, we offer 3 capping and provides 97% of the capping efficiency. And for the uridine modification, we also provide different options. And for impurity, we optimize our product producing process by usually the way for purification is oligo-DT to and the liquid nanoparticle packaging is the last step to package the mRNA. We also give you different options. And this is the whole workflow. We can give you one-stop solution. So, and the manufacturer manufacturer location is in Seattle. Okay, so this is the about the mRNA key considerations. If we look at some details, we dig out more. First is about the linearization of the DNA template. So linearization is necessary for in vitro transcription. So we make it, we use a restriction enzyme to make it from single to linearized plasmid. Because if we still use circular DNA, it's like no end for transcription. So the length of the mRNA is different. And so it means we will get unnecessary impurities. But for linearized DNA, it can ensure the transcribed RNA of defined length and sequence. And why do we have to outsource the plasmid DNA from GenScript? Because here's a comparison between self-made linearization versus the linearized by GenScript. We can see that self-made linearization has the obvious miscellaneous bands. But this one is more clear, provided from GenScript. And about the poly-A tail part. So the length of poly-A tail is the important evaluation. Poly-A tail can promote translation and prevent mRNA from degradation. And usually the length of poly-A tail should be more than 80. And I just mentioned our way to tailing it is to use a plasmid. We put poly-A tail cloned in the plasmid template. Okay, so here shows the different lengths of the poly-A tail and how the length impacts mRNA expression. And okay, so next is about the capping. So we provide 3 kinds of capping: cap 0, cap 1, or cap 2. And our efficiency of capping confirmed by this LCMS is 97%. So it's quite efficient capping. And next element is about the nucleotide modification. So why do we have to modify nucleotides? Because it can reduce the immunogenicity of exogenous mRNA and also can enhance mRNA stability and also can improve the translation efficiency of mRNA. And Moderna and BioNTech developed modified mRNA products that are more than 90% effective at preventing infections. And the leading unmodified mRNA vaccine was 47% effective. And we'll show you later that GenScript provides a variety of chemically modified MPT options. So when we have this mRNA, and the purification process is also very important. So for crude purity, we use oligo-DT affinity chromatography to remove the protein residues and small molecules. And we use this hydrophobic exchange and other methods to remove the dsRNA. So here shows you what a high quality IVT mRNA should like, and here's the RQC. Okay, so GenScript provides the one-stop solution for mRNA service, so we can start from codon optimization design, and then gene synthesis, then cloning, and the plasmid preparation. And then in the meantime, we will also provide some mRNA production related enzymes. And then after linearization of the plasmid, we provide IVT mRNA synthesis and also the circular mRNA. And the last step is the lipid nanoparticle package. So if you just send us your ORF sequence, we will design your mRNA expression vector with all these important elements. And then just we will just start right away from gene synthesis to linear plasmid, then IVT mRNA production. And here shows you what options we provide for the key parts of the production, mRNA production. And for the circular RNA service package, you can see the options, and we have the very fast delivery. The turnaround time is within 3 weeks. Okay, we talked about mRNA and also compared mRNA and circular RNA and saRNA. Now let's spend some time to see our proprietary circular RNA platform. So compared with the published method of producing circular RNA, GenScript have our own proprietary circular RNA production platform, and this platform shows improved circularization efficacy and also increased purity. And the optimization of the circular RNA preparation method can lead to increased protein expression later compared to the linear mRNA and also the published method. And what the key advantages of circular RNA platform comparing to mRNA? There are 3 advantages. The first one is increased and prolonged protein expression. Here is results. Protein expression level post-transfection with linear IVT mRNA remained stable, but the circular RNA exhibit increasing protein expression level over the course of 3 days and resulting about 2.5 folds higher expression per well than mRNA. So it's about the protein expression. And also, circular RNA shows increased and prolonged half-life compared to mRNA. We can take a look at these data. Slower clearance or degradation of circular EGFP RNA was observed compared to linear mRNA. And it's a modified version. And equimolar amounts of circular or linear EGFP RNA were transfected into A549 cells, and about 2- and 4-fold higher circular RNA were detected in 2 to 3 days. And the last advantages is the lower immunogenicity compared to Cap1 mRNA. So here's the circular RNA in different in A549 cell, and we tested the cytokines expression. So the less cytokine expression means the lower immunogenicity. And the gray one is Cap1 mRNA. So after the mRNA is produced, we need the last step, which is the LNP package. So here's the option for our services, because different mRNA applications need different delivery vehicles, and we're providing you tailored LNP formulations for your vaccine delivery strategies. Okay, we talked about mRNA, and now let's take a look at the importance of neoantigen peptide for personalized cancer vaccines. So, talking about personalized cancer vaccine, first, we should identify the tumor antigen. And talking about personalized, it means the antigen is the TSA, tumor specific antigen, and the new antigen is one kind of TSA. So, and one important part of TSA. So when we talk about personalized cancer vaccines, usually what we mean is the antigen of the tumor is new antigen. So we're trying to find the more new antigen, the better. But no, after we see all these failures, we think improving hit rate may be the key. So we can see the cancer vaccine candidates all failed in clinical study. So for now, there's still no successful case launched. And the main reasons for clinical trial failure, we think one of the reasons is that it's difficult for the human body to generate an immune response to many new antigens. And even if an immune response is successfully triggered, cancer cells may still escape. So from this, we can realize that screening of new antigens and combined drug maybe it should be the general trend in a future development of tumor vaccines. So let's see the screening of new antigens first. And the importance of using new antigen peptides when screening and validate tumor new antigens. So we usually now with the development of technology, and also we need to do the immunogenicity test later. And the purpose is to screen from like tens of thousands of new antigens to find the validated one. So maybe only here shows like 3 were validated finally. So during this process, screening and validation process, an important material is new antigen peptides because we need the peptides library or a mixture of the new antigen peptides to incubate with the PBMC. Then we'll see whether the cytokines can be produced, which means it shows the immunogenicity and means the new antigen is the one that we want. So new antigen peptides is an important material. And GenScript is providing you the total solutions for the neoantigen peptides screening and validation. First, we have the peptide library design tools. So there are different kinds of libraries, and there's an online design tool you can use and to design your peptides. And also there's another tool to assess the difficulty of the peptide synthesis. And regarding to the peptide library synthesis, we have this automatic platform. It's high throughput, and we can produce up to 600 peptides at the same time. And regarding to the quality control, we can guarantee 100% of the all the new antigens peptides by this acute pooling service. And why we have to guarantee this because it's a peptide mixture or peptide library, right? And we should guarantee all the peptides produced to avoid the reduction of the epitope screening efficiency caused by the deletion of the neoantigen peptide sequences. And also we control the production environment. It's a grade C clean room to avoid the contamination. So that's about the tumor antigen discovery. After we have identified the new antigen, the next step is to produce the vaccine. And here shows you the considerations relating to personalized cancer vaccine. So there are several kinds of vaccine formats that you can choose, and it's a personalized vaccine. So the patient is waiting for us. So the time of manufacture is very important to be evaluated, and also the vaccine dose, like do you need like a booster dose or like after the disease recurrence, whether you still need one more dose, stuff like that. So vaccine dose is another consideration. And also I mentioned previously, in order to have a success treatment, the combination therapies is also important effect. And we should think about like pre-vaccination using some cytokines to promote baseline immunity, and also use the immune checkpoint inhibitors to boost vaccine-induced immune responses. So during the production of the personalized new antigen cancer vaccines, we also need new antigen peptides, and it plays an important role for the development. For example, here shows you different vaccine types. And if you are developing peptide vaccine, and of course, peptide itself is be used as active ingredients. So it's directly used as drug substance or drug products. And if you are developing mRNA vaccine or DNA vaccine, new antigen peptides can serve as positive controls for vaccine screening and immunogenicity verification, and also for and DC vaccine, the peptides is the crucial raw antigen material, which will be used to incubate with the dendritic cell. To preload we need to preload the new antigens on DC cells, so it can pass the signal to T cell for the stimulation and later tumor cell destruction. Now we know the importance of new antigens for the personalized cancer vaccine development, but actually the peptide itself is not easy to be produced. These are the considerations that making it difficult to produce. One is the length. New antigen peptides usually range from 8 to 50 amino acids, but peptides, if it's longer than 20 amino acids, it's difficult to synthesize. And also the charge will affect the purity, and also the hydrophobicity also affects the purity, and making the synthesis harder, because it's hard to soluble in water. And the yield, it's very difficult to generate a very large amount. Okay, that's the evaluation when we produce the peptides. And in order to solve this, GenScript has the streamlined workflow, starting from the sequence analysis for fasting software to peptide synthesis and purification. We have these two platforms to guarantee more than 98% synthesis success rate, and the high quality delivery could be as fast as 1 week. Now this is the case from BioNTech. It's published last year in May, and it's a very important new antigen vaccines that many people are paying attention to the development of this vaccine. I think right now it's in clinical phase 2. And this vaccine, it uses GenScript new antigens for in vitro stimulation and T cell clone specificity to peptides. Let's take a quick look at what it is about. So it's a personalized RNA vaccine, and the target is new antigen. We're trying to use this kind of vaccine to stimulate T cells in pancreatic cancer. So first we get the tumor, and then we transport the tissue and sequence the tumor and normal DNA, trying to, like I mentioned, using AI to technology to predict and finally select the new antigens. Then we send it to the manufacturer to produce the customized mRNA vaccine, which includes the neoantigens, and it's packaged by lipoplexes. So this is the personalized mRNA. When we produce it, actually, we are in the meantime we give this patient the anti-PD-L1 treatment. Then we have the first dose of the vaccine. Later, there's a 4-drug chemotherapy treatment. Then we give this patient another booster dose of the vaccine. So that's the general procedure. That's about this case. Okay, so last part is overview of GenScript vaccine development workflow and solutions. We can support you from early research to clinical trials. Today we talked about tumor study, but also we can support your pathogen study for the infectious disease protection. These are all related services we can provide during your antigen discovery. After the antigen is identified, the next step is the vaccine sequence design and modification and preparation. You can choose different types of vaccines, and we can support you from gene synthesis to plasmid preparation, either then to IVT mRNA production, or you can choose different cell types for protein expression. Or if you want to use viral vectors, we have AAV vector one-stop solution and lentiviral vector one-stop solution. And of course, I mentioned peptide synthesis today. So we have peptide synthesis and also neoantigen peptide synthesis. And for the vaccine effect evaluation part, for like DNA or mRNA vaccines, we need to test the antigen expression. We have some popular products like precast gels, and these two are instruments for protein transfer system and also Western blotting. And for cellular immunity evaluation, of course, you can also use our peptide pools, which are customer service, and also for the cytokine detection, we are supporting you antibody development. And for humoral immunity evaluation, we have the ELISA kit generation service, and also for these SARS-CoV-2, we have the ready-to-use product, including the pseudovirus neutralization assay kit, and also this CPAS SARS-CoV-2 neutralization antibody detection kit. And for the clinical development, we have GMP plasmid DNA manufacturing, and also GMP biologics. And in the meantime, we are providing some ELISA kits or endotoxin detection kits for your QC analysis. And we can provide more, and just contact us for details. And thank you for your time, and that's all for my presentation.
Thank you, Melody, for your very informative presentation. We will now start the Q&A portion of the webinar. If you have a question that you would like to ask, please do so now. Just click on the Ask a Question box located at the bottom of your screen. Let's get started. Okay, the first question we have here is, can I use plasmids I have already made with GenScript in previous gene synthesis orders for mRNA production?
Yes, of course. We can just subclone your ORF sequence into our proprietary vector, and this vector contains T7 promoter, UTR, and 100 poly-A tails for the subsequent mRNA production. Yes.
Wonderful, thank you. We have one more question here. Is circRNA production time the same as mRNA?
Not exactly. It's actually a little bit longer than mRNA. One of the main reasons is that there's an extra step of circularization for circular RNA. And another reason is that we use our proprietary purification method, which is HPLC method. So this purification process is going to be a little bit longer. So your circRNA is a little bit longer than mRNA production. Yeah.
Perfect, thank you. It looks like we have time for one final question. I might use cGMP neoantigen peptides later. Do you have cGMP grade peptides? What are the differences between RUO and cGMP peptides?
Yes, we do have cGMP grade peptides for the preclinical study or further clinical study. And the differences between RUO grade and cGMP grade, generally speaking, is first, the environment is different. For RUO grade, usually they are produced in a controlled but unclassified facility. But for cGMP peptide, they are produced under grade C environment with grade A isolator. And another difference is the quality management system difference. So for RUO grade, usually it's produced under ISO 9001. I think it's kind of well known. But for cGMP peptides, the quality management system is called ICH Q7A. And there are also other guidances for GMP peptides. You can contact us later for detailed difference comparison. Thank you.
Awesome, thank you so much. All right, well, thank you again, Melody Chang, for your time today and for your very important research. Please be sure to also check out GenScript's booth located in the exhibit hall for more great information. Before we go, I would like to thank the audience for joining us today and for your very interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.